Hello and welcome. Today I wanted to continue the video that I started about nature's design, that how cells have been designed by nature and engineered by evolution. I talked a lot about membranes. I talked about how different organelles uh, uh, evolved inside the cell by various means to perform different kind of function in different separate environments. So purpose of evolution of this separate organelles is just to ensure that a particular kind of reaction happens in a particular uh, uh, individual dedicated environment. So, so I talked about mitochondria, that how different proteins carrying different kind of atoms and metals are located on the membrane of it to perform various kind of function. I talked about other organelles like lysosome and Golgi body. One thing that I did not talk, and I promised you to talk in next video, and hence I thought to finish it, and that thing is about nucleus, that what is happening in the nucleus. So as I told you that, and you perhaps all know, that nucleus is a house for the genetic material. So and in our cell, so when I talk, generally you consider I'm talking about my cell and you cell if you are a human and you're looking at me. I don't know if this video, if some alien would see this video, I don't know what would they think, but alien is an alien concept, and let's stick to self concept, that inside, take out any cell of my body, put it on this microscope and look at the magnified view of cell, you would realize that there is a big nucleus at the center of the cell, and maybe I can show you out here on the screen. Right now I am with Henrietta Lack's cell, and her cells too have nucleus, but the only problem with showing nucleus is just to make you look at the whether resolution is sufficient enough or not to visualize your nucleus from the cell. But I'll show you. Before that, I would like to continue of the argument that what kind of engineering nature have done to preserve the information. So see, everything works as per some prior information. So our function, I'm speaking, and in order to speak something, my larynx are working, and to make the larynx functional, larynx required a lot of protein, and different proteins are different kind depending on their sequence, and these protein workers, because of these protein workers, our phenotype comes. So whatever we are today, whatever our behavior is, whatever our action is, everything is happening at the bottom level by protein workers. So these protein workers perform various kind of work inside the cell to perform a particular kind of function or any kind of function. And so different kind of protein does different kind of function depending on the sequence of amino acid in the protein. So if protein A is protein A and this is protein B, and we are calling it a different protein. So the why are we calling different protein different is essentially because of the sequence of amino acids are different. And if sequence of amino acids in protein A and protein B are different, it essentially means that it would fold into different shape, different structure. So structure of a protein, whether it will be like this, or it will be like this, or it will be like this, or it will be like this. So it essentially depends on the sequence of amino acid in the protein. So sequence of amino acid is the information that decide the structure of the protein and that decide the function of the protein. But who decides sequence of amino acid in the protein? So there should be a map on which design sequence of amino acid in the protein, and today we'll talk about that map. So as I told you that nature have made a really well defined boundary in eukaryotic cells, our cells that we call nucleus, and we have genetic material in that nucleus. So the information for a particular sequence of amino acid in a particular kind of protein is essentially inside the nucleus. That information is present in the genetic material, and in our case the genetic material is DNA. So what is DNA? So DNA is again a big chain or polymer of what? Polymer of nucleotide. So various nucleotides attach with each other and forms a stretch, a long chain of something, a long chain of that nucleotide, and that we call DNA. So as you are all aware of the fact that see, protein molecules, since I started my series from protein only, so you must be knowing that protein molecules are series of amino acids, and there are 20 kinds of typical amino acids, 1 or 2 or maybe 3, because some people consider water an essential partner in deciding shape of protein, and then they advocate that water should also be called as the 21st amino acid. But this is just to ensure the importance of water in the cytoplasm that decide the structure of protein, which is very essential. But if you look at the sequence of amino acid in the protein, it's only 20 kind of amino acids, and 2 atypical amino acids are also there, which is present in some systems, some organisms, in some species. If we talk about DNA, then DNA is again a polymer of nucleotide, but nucleotide in DNA is only 4 kind of nucleotide. Okay, and when I say nucleotide, you should understand nucleotide is also a compound. Okay, so when you look at the structure of nucleotide, you can Google it and you can look at the structure of nucleotide. So nucleotide has one nitrogenous base. So nitrogenous base is of 4 kind. Okay, adenine, thymine, guanine, cytosine. So for this video, I'll keep talking like A, T, G, C, adenine, thymine, guanine, and cytosine. Now, so with this nucleotide, there is a phosphate sugar attached to it. When I say phosphate sugar, it's a monomer, monosaccharide made up of 5 carbon, and this phosphate sugar, which takes a ring-like shape when you put a sugar monosaccharide in the water, or disaccharide in the water. Sugar essentially takes a ring-like structure, and just to gain stability, I'll not talk about that. So there are 4 nucleotides, and with each nucleotides a sugar is attached. Okay, and again with this one more group is attached, that is the phosphate group, and this whole structure, sugar, nitrogenous base, and phosphate, we call this whole compound as a nucleotide. If you leave phosphate, only sugar and nitrogenous base, then it is called nucleoside. But who cares about nomenclature? Yes, if you are preparing for some competitive exams, and if there is somebody who are out there to judge you based on whether you know the nomenclature or not, then you must remember all this nomenclature and you must refer to your textbook. However, if you are talking to me, I am least bothered about nomenclature, but I know the nomenclature. So essentially what I am saying there, depending on 4 kind of nitrogenous bases, adenine, thymine, guanine, and cytosine, there are 4 kind of nucleotide. So if I am not wrong, the nomenclature is like adenosine, thymosin, guanosine, and cytosine. That's correct. Let's take it. So I'm telling this 4 kind of compound, which is 4 kind of nucleotide made up of 4 different kind of nitrogenous bases, are linked by each other with covalent bond and form a long polysaccharide, long polymer, and that long polymer of nucleotide we call DNA here, which resides inside your nucleus. So Uh, and if we talk about our DNA, then our DNA, which is a eukaryotic DNA, is double-stranded DNA. Means there is a long chain of polymer of nucleotide attached with each other by covalent bond, making one chain, and there is another polymer of nucleotide making another polymer, and they just make a double-stranded chain. They just sort of associate with each other. This chain and this chain will associate with each other. I'm talking about structure of DNA, and they form double-stranded structure. Now, when I ask you how different nucleotides are attached with each other in one strand, you would call bicovalent bond. And if I ask you, and this chain is also a polymer of nucleotide attached with each other by covalent bonds. Now, if I ask you that how these two strands are bonded with each other, then you would call by hydrogen bonding. So these two double-stranded chain. So why these bondings are important? Because it is the bond that makes something stable. So bond is not a straight line. I explained it multiple times at various places. But I must tell you that bonds are not lines. Bonds are essentially a way to tell in very short that, see, these things have done this kind of overlapping, and because of this kind of overlapping, two molecules meet with each other and remains stable. So whenever bond forms, there is a release of some energy. When covalent bond forms, then huge release of energy. And if there is a huge loss of energy to the surrounding, there is a huge gain of entropy in the surrounding molecules. And that will make the process very much stable, because take it as a rule of thumb, more you increase the entropy of the universe, more stable that system would be. So whenever bond forms, there is a release of heat. That heat increases the randomness of the surrounding, and because of that, this formation of bond, this interaction or bond formation makes this process, makes this compound. Let's say if these are two different, my fished are two different atoms making a bond, then this system will be more stable as compared to when they are apart, because they have released some heat to increase the entropy of the surrounding. And second thing, so if I ask you that, let's say if it is a covalent bond, then formation of covalent bond would release more heat, that would increase the entropy of the surrounding more. If it is some hydrogen bonding, which happens because of partial electrostatic charge present on the atom, which is again associated with an atom with different electronegativity. I'll not go in detail. I want to go. It's just that I'm thinking about my experiment. Light will go and I'll start my experiment, and I also don't want to go to a place where I cannot return back from, because these are my interesting these are the interesting field. And second of all, I just realized that I keep on advocating that words doesn't matter, nomenclature doesn't matter, but nomenclature perhaps has made to say things in a precise manner. Like every time when I say bond, you should understand what do I mean. I should not keep on explaining that bond means this. So that's why nomenclature mean important, and that's why kids at home should be knowing the nomenclature. But when you hear that word, that particular nomenclature, you should immediately understand their meaning. So if I call bond, then you should be thinking that there is some interaction. Because of that interaction, there is a loss of heat that makes these separate molecules more stable when they are nearby. In the same way, one strand of DNA and other strand of DNA adhere with each other by hydrogen bonding. and every nucleotide in one strand and every nucleotide in another strand are adhered with each other by covalent bond. So this is more stable as compared to the bond between two strands. So if you, let's say you have isolated some DNA from my cell, you can isolate it, right? You just have to throw all other things. It's just like sheaving, and there is a method called chromatography by which you can separate one particular macromolecule or anything of your interest from other things. So let's say you have collected your purified DNA from my body, my flesh, and you are boiling the DNA in 100 degrees centigrade water. What do you think? Would nucleotides which are attached with each other by covalent bond, would they break? No, they wouldn't break, because bond energy of covalent bond is very high. Even if you boil your DNA at 100 degrees centigrade, covalent bond would not break. But the hydrogen bond that are present between strand 1 and strand 2, they will definitely break, because their binding energy is less, and you would see two strands of DNA would go away from each other. But at normal temperature, at normal 37 degrees centigrade, your two polymer, two strands of DNA will remain intact with each other, and it would remain in association with each other and form double-stranded DNA. Now, you take any cell of mine, look at in the microscope, and you would see if resolution and magnification is sufficient, you would see this DNA molecules are very dynamic molecules. They take different shape, even though their basic shape is double-stranded DNA, but they, as per the requirement, they keep on doing random stuff. Like if it is a time of cell division, then DNA molecules wrap around the protein molecules, which are histone protein, which are non-histone proteins, and they form some structure that we call chromosome. So chromosome is nothing but when DNA wrap around histone protein, and again this structure that we call nucleosome, this structure again meet with other proteins and form a condensed small and in fact structure that we call chromosome. And when they are active, when DNA are active, what do I mean by DNA are active? When DNA are doing their function. And what is the function of DNA? The function of DNA is to give the information. One function of DNA is just to give the information to code for a particular sequence of amino acid in the protein. So when a DNA has to do this, then DNA would not like to remain in chromosome. Rather, they would like to remain free from other proteins so that this process can happen. And now I should be explaining what process I am talking about, that how nature designed DNA in a way that information for the sequence of amino acid in the protein get transferred, get implemented. So I again want you to know that why I keep telling you that DNA has information for the sequence of amino acid in the protein. And why is it important? Because different protein, our living system, and any living system for the matter of fact, works because of presence of different kind of protein. If you have to just let's say transfer oxygen molecules from lungs to different cells to perform respiration reaction, then you require a particular kind of protein molecules that is called hemoglobin. If you want, as soon as you have eaten something and digestion happened and glucose molecule absorbed in the blood, now you want this glucose molecule to go inside the cell so that respiration process can happen. And in order to push the glucose molecule from outside to inside, you require a protein molecule that we call the insulin. In the same way, we have glucagon. if you are in a if you are living in a African continent where there is so much of heat, then you would like more melatonin protein or melanin protein. There is a slight difference, but melanin protein to form on your skin, so that your body system does not get affected by sunlight, and you would want more melanin protein to form in your skin. And on the contrary, if you are in some cold country, then you would want you would want that less melanin to form in your skin. And that's why that's what we see. Like if you go Africa, you will see everybody is black because they require more melanin to survive in that kind of environment. And if you come here in cold country, we have less melanin in our skin to which is essentially not needed here in cold country. Now but India seems, because of colonialism and a lot of other things, India is a mixture of everything, and that's what make it democratic. Or maybe ideology is very much democratic. Ideology of India is very much democratic, and we are the largest democracy, and we all should proud of on it, even though other people try to manipulate you against your own country. Don't get manipulated. We are from a very we should proud of our country, and we should because of logic and because of reason. If you look at the history of India, you would realize why should we be proud of being in this country. Having said that, so different as per different requirement, we require different protein. Now the question is, who decide which kind of protein would form in your skin or in your body? Everything that happens in your system is happening because of different kind of protein. And why different kind of proteins are different? Because of sequence of amino acids in the proteins are different. Now who decides the sequence of amino acid in the protein? It is the DNA and the sequence of nucleotide on the DNA. So when DNA is in active state, so DNA is a very long stretch, and about few percent of your DNA macromolecule has codes. Like I would say, if I'm not wrong, it's around 1% of DNA that you're housing in each of your nucleus have code to sequence for to have code to provide information to code for a particular kind of protein or messenger RNA, and this part of DNA is called gene. So very small portion in the gene in the DNA which have information in the form of sequence of nucleotide to code for a particular kind of protein or particular kind of messenger RNA, and those stretches of DNA are called gene. Okay. I think I should stay some more time here to understand DNA macromolecule more, and then I'll proceed that how the information present in DNA in the form of sequence of ATGC, which are nucleotide, actually codes for sequence of amino acid in the protein. So okay, so let's understand DNA more. So if I ask you what is RAM, so it is RAM, and if I ask you what is MAR, it is MAR. MAR essentially means beating in Hindi. So meaning completely changed. So when you say RAM, then it's a ideal picture of somebody who does only right things. And if I say MAR, then it would be different. So you have seen that sequence of alphabet matters. However, I don't advocate a lot about again which word we should choose to do which kind of function. But But it is important to give this analogy that sequence of alphabet changes. If you change the sequence of alphabet, it changes the word. In the same way, this is the ideology of literature people, and our DNA evolved way before than this RAM system or language system have developed. And during those time when engineering was happening, perhaps nature selected this way to store information in the form of sequence. So even if you read a book, so book is a source of information, and that source of information is there in the form of sequence of words, and then sequence of words are again joined by sequence of words, and then sequence of alphabet joined by sequence of words, and then statements, and then further information is stored in the form of whole book. In the same way, in your DNA we have sequence of different nucleotide, and these different nucleotide essentially code for sequence of protein. But how? So now I should go in detail. Okay, so if you look at the cells when they are not dividing, so to understand dynamics of DNA in your nucleus, we need to look at basically two things: a dividing cells and a non-dividing cells. So first let's talk about non-dividing cells. Your cells are not dividing, and it is doing its function. Maybe in your brain you have neurons, and inside your neurons you have a nucleus, and your nucleus has let's say DNA, and we have taken out your neurons, we have put it on microscope, and we are looking it through this eyepiece, or maybe through this. And if your microscope has good resolution and magnification, then you would see DNA, and you cannot see the sequence of nucleotide in the DNA using this microscope. That requires another technique to understand. But what you would see, that nucleus are all all the nucleus are in condensed form. They are like thread, and those condensed form of nucleus. So when I say condensed form, I shouldn't say condensed form. They are all just meshed up. And they are free. They are not bound to any protein, but they are protein acting all the time. So you would see you would see the DNA is making your double-stranded DNA and freely lies in your nucleus. Nucleus is essentially some 70-60% water and other ion molecules, and then your DNA is dissolved in that water of your nucleus, that we call nucleoplasm. Now one more point here is what happens, that your DNA has a sequence of nucleotide in the form of ATGC, and then another protein worker comes there, okay, and one protein worker comes there, and they just bring another protein workers, and then there are multiple, maybe hundreds of protein workers. They come and they read the sequence of nucleotide ATGC on the DNA, and after reading ATGC on DNA, they form a complementary molecule, and that complementary molecule is called RNA. And here is messenger RNA, because all I am talking in this video is how who decides sequence of amino acid in the protein. And I said that code for sequence of amino acid in the protein is essentially on the DNA in the form of sequence of nucleotide, but DNA never goes out to form protein on ribosome. You must be aware that protein synthesis happens on the ribosome, and for this process to happen, information has to come out of nucleus and go to the ribosome. So your main information is DNA, which stores in a completely protected environment that we call nucleus, but the information has to go to the ribosome. And who brings information in the form of sequence of nucleotide which is present on the DNA. So who brings this information of sequence of nucleotide on the DNA to ribosome, where sequence of amino acid in the protein will be decided, and the answer is messenger RNA. So what happens when you are looking at your neuron cells which are no more dividing, and you would see that the DNA molecules, double-stranded DNA molecules are lying there in the nucleoplasm environment, which is essentially 60-70% water only, and there are multiple protein workers are working on the DNA, and the size of these DNA and protein molecules are in nanometer range. So they are the size of protein is nanometer, or maybe some micrometer if it goes high, but never. I wouldn't come, because typical size of a protein is typical size of a cell is around 20 micrometer. So I would say essentially all the protein molecules are in the nanometer size scale. Having said that, so on your resting cells in your neurons, you have DNA, and there are multiple protein partners who are working on the DNA, and they work on a DNA in a particular way to form different molecule, and that different molecule is messenger RNA. So messenger RNA is also sequence of nucleotide only. In the same way, DNA is also the sequence of nucleotide. Only difference is messenger RNA do not have thymine. Instead of thymine, they have uracil. So if I ask you what is the sequence of DNA, then you would call it is a random sequence of A, T, G, C. If I ask you what should be the sequence of messenger RNA, then it is also the sequence of nucleotide, but what all nucleotides are present on messenger RNA? It's adenine. So I'm talking about nitrogen species: adenine, uracil, not thymine, uracil, guanine, and cytosine. And you know that so light is gone. So I'll set up some reactions, and then I'll come back to make you understand that or maybe I'll finish it up, that this sequence of AUGC on messenger RNA is decided by sequence of ATGC on DNA. I think I should just switch to some mode, and then I'll continue, and then I'll talk about how this thing messenger RNA brings information from DNA to the protein, and then they do certain kind of function. So give me 1 second. Okay, so multiple protein molecules which are present in the nucleus, and they have ability to understand the sequence of nucleotide on DNA, and somehow tell this to the messenger RNA, and somehow transfer it to the messenger RNA. So friends, okay, so using this protein machinery, the information of nucleotide present on DNA, sequence of nucleotide present on DNA is copied to the sequence of nucleotide in messenger RNA, and this process happens inside the nucleus, and researchers named this process transcription. So transcription is essentially the copying of sequence of nucleotide on DNA to sequence of nucleotide on messenger RNA. Having said that, now you see that this sequence of information, this information is on messenger RNA, and then messenger RNA leaves the nucleus. It comes out of nucleus and goes into the cytoplasm. And so you see, DNA is very important thing. It has the basic codes for for sequencing amino acid in the protein. And since nucleus, this DNA is very important, they have the main code for any protein formation. That's why nature had to keep it in a very protected environment where everything cannot access. So nucleus, these DNA molecules are in the double membranous structure that we call nuclear membrane, and there are small pores outside the nuclear membrane, and that pores are called nuclear pore, and inside it your DNA molecules lie, who do not come out to do some function, because you never know if it is come out in the cytoplasm, what can happen. So nature had to keep all DNA inside the nucleus so that nothing happens to them. And how would then work happen? Nature designed and evolution engineered it in a way that messenger RNA will leave the nucleus carrying the information. So now theory of DNA is over here, but I'll come back again, because right now I am talking about a non-dividing cells. I am looking at cells of my neurons. I'll come and at the end I'll come to another part, what happens to these DNA molecules when they are in a dividing state. But for a non-dividing cells, so work of DNA is just using some protein machinery. The information present on DNA is copied to messenger RNA in the form of AUGC, which are again the nucleotide. Now this messenger RNA comes out of the nucleus, and it will wrap around the ribosome because of certain design, because using some kind of protein. Now what happens, there is another kind of RNA, and these RNA are called transfer RNA. So every cells have multiple transfer RNA, and if you look at the structure of transfer RNA, it's like a clove leaf, and in folded structure, some L-shaped structure. So essentially you see this is your transfer RNA. So in this transfer RNA, what is there? At one hand you have amino acid. In the transfer RNA you have amino acid, okay? And different transfer RNA has different kind of amino acids. Some transfer RNA is made to bind with glycine, some are made to bind with alanine, some are made to bind with proline, some are made to bind with carboxylic acid, the let's say glutamate or aspartate. Some are made to bond lysine, some are made to bond arginine, some transfer RNA are made to bind cysteine, and so on. Okay, and another side of it has some sequence of nucleotide. If all the DNA and all the RNA are the polymer of these nucleotides. So what is there? So this hand of your transfer RNA is bind to different kind of amino acid, and which kind of amino acid would it bind to is totally that information remains here as well in the sequence of these three nucleotide. So a transfer RNA, if you consider transfer RNA has two hands, although it has other loops, D C loop and other loops as well. You look at the structure of transfer RNA, it's clover leaf structure it has, but it is always bind to in one hand by some amino acid, and on other side it has a sequence of amino acid that we call anticodon. What do we call it? Anticodon. Okay, so when we talk about anticodon, then we should define codon as well. So as you remember, the messenger RNA, which has information present on DNA in forms of sequence of AUGC, has come out of the nucleus, and it is on the ribosome. Now these the sequence of nucleotide on messenger RNA codes for the sequence of amino acid in the protein. How? So nature has designed and selected it in a way that each three amino acid, each sequence of three of nucleotide in messenger RNA is termed as codon. Okay, and this sequence of three nucleotide on messenger RNA actually codes. Is the genetic code actually codes for the sequence of amino acid in the protein. How? So your messenger RNA has multiple sequence of three nucleotide that we call codon. So when I say codon, you understand the sequence of three amino acid. The sequence of when I say codon, you should understand sequence of three nucleotide on messenger RNA. So there are multiple codons on the messenger RNA, and each codon, now I will not say sequence of three amino acid. I would not say sequence of three nucleotide. I would say that one codon. This is one codon. Okay, and there are multiple codons present on messenger RNA, and each codon codes for one amino acid. Each codon code for one amino acid on the protein. How? Because sequence of I'll start from the beginning. Sequence of nucleotide, which is the genetic information present on DNA, is copied to messenger RNA using some protein. Now sequence of nucleotide in messenger RNA will be given to protein using one molecule that we call transfer RNA molecule. So these transfer RNA molecules are basically the carrier molecule, which has amino acid in one hand and anticodon in another hand, if you look at the structure of transfer RNA. So at the ribosome, what happens that these transfer RNA reads the sequence of nucleotide that we call codon present on messenger RNA using their anticodon. So transfer RNA has anticodon, and using this anticodon they read codon sequence on messenger RNA. And codon sequence on messenger RNA, so there are multiple transfer RNA, and let's say you have a code for glycine on your messenger RNA. Then glycine carrying amino acid would come with its anticodon, and they would sit on the messenger RNA that has that particular codon, and reading that particular codon, that amino acid will be transferred from transfer RNA to the ongoing synthesizing protein. So protein synthesis happens. First let's say first codon is AUG, or maybe UAA or UGA. Starting codon can be 3: UGA, UAA or UAG. Okay, 3 of them. So as soon as this is the first sequence, then a transfer RNA would come who will read this codon using their anticodon and put their protein there. Let's say methionine. They have put methionine out there because one arm of his has amino acid. Then next and as soon as they have given amino acid, they would leave the place, and then next transfer RNA would come who will read the next codon and add next amino acid. The next transfer RNA would come who would read the next codon using their anticodon and give that particular kind of amino acid. They will leave. Next transfer RNA will come. They will read next codon using their anticodon and give another amino acid. And this process keep on happening till stop codon doesn't come. So your messenger RNA has stop codon as well, it has start codon as well, so that very precisely and very accurately sequence of amino acid can be coded in the synthesizing protein. So, and this whole process is called central dogma, that how information present on DNA first copied into messenger RNA using various protein, and we call this whole process as transcription. And messenger RNA then comes out of your nucleus, goes to the ribosome, and then transfer RNA comes. Transfer RNA has anticodon, and they read codons, which is the sequence of 3 nucleotides present on messenger RNA. They read codon on messenger RNA using their anticodon, and add that particular amino acid to the protein, and then protein synthesis happens. And after synthesizing, protein folds, and even co-translational folding also happens. During the synthesis, folding happens, and then depending on the sequence of amino acid in the protein, a particular shape is taken by the protein, and protein behaves and function as per their structure. So this is how different functions in our body happens, and this is what nature has designed, and this is the design of nature, selected by natural selection, nature itself, and that we call evolution. The engineering is done by evolution. As I said, that I'll first talk about non-dividing cells, and then I'll talk about dividing cells, and then I'll conclude the video. So the point I want to say that if you look at some dividing cells, let's say you have got a wound somewhere. Okay, I'm giving you this example just to make sure that you understand what does dividing cell mean. Otherwise, all of your cells, most of your cells, keep on dividing in a cyclical pattern. So every cell goes through different cycle, and then they divide as per the requirement. So if you think that there is a cell, resting cells, and it has nucleus, and nucleus has a lot of DNA making thread-like structure, would it be easy to divide the DNA into different cells accurately and precisely? I think you would answer no. It's not easy to divide this DNA into different cells very accurately and precisely. Then what should be done? So how do we work? Like just to stitch your cloth, you need this thread, and then when we go to buy thread, then what do? We simply don't buy thread in a messed up way. We have thread wrapped around a piece of paper, and then we have multiple that kind of structure. If you remember, we call it reel. I don't know, what do we call it? So threads are generally wrapped on some paper, some wood, or some wooden structure, and that's what they sell it, so that there should be a quantification of what is being sold. So in a same way, nature also designed the cell division process, that no, we will not divide DNA molecules, which are the genetic material, into cells in a form of some thread. So what do? They first, if you have to, if a cell has to go through cell division, then first of all, cell has to duplicate the number of duplicate their DNA content first of all. So what happens? That is called synthetic phase. If you have to go through, you want to go through, so it works like this. If a cell has to go through cell division, first cell has to duplicate all of its DNA content. Okay, so that is called replication. So there is so in this process of replication, exact copy of DNA forms. The exact copy of DNA forms in your nucleus. Now this is important to understand, that who make this replication process possible? All these protein workers. Any work that happens inside the cell are mostly, and perhaps I would no hesitation to say that it happens all the time in using some protein worker. So proteins are worker inside the cell. In using these protein workers, dividing cells first divide all of its DNA in S phase, we call it S phase of cell cycle, and that process is called replication of DNA. Now what happens, these DNA gets condensed and they gets wrapped around the protein molecules. And what are these protein molecules? Some histone molecules and some non-histone molecules. And when DNA wrapped around, first they form nucleosome and they are also condensed, and then they form chromosome. So when DNA first wrapped around histone and then they are called nucleosome, four histones, and it is forming some wrapping around the protein, and these nucleosomes again wrapped around different kind of other proteins, and then they form a condensed short and fat molecule that we call chromosome. So if it is if you look at the chromosome after S phase, where DNA replication has happened, then you would see that every chromosome has two replicated same thing, and that we called the sister chromatid. This chromatid and this chromatid is sister chromatid. So it should be, I don't know how to show it, but it should be like this. Essentially, I think I should switch to some other things where I can show you how should it look like. So it should you consider everything is same. So this would be the structure of your stuff. Okay. So you have, every chromosome has two chromatid, one chromatid this side and one chromatid this side, and this thing that you see is called centromere, or which has again some other protein layer that we call kinetochore. Friends, one more thing that I must mention, that if you are looking at your somatic cells like skin cells, every cells in your body has two copy of DNA, two copy of chromosome, one that has taken from mother and one that has taken from father. So how do we form? We form from two cells, one cells from mother and one cells from father, and then they fertilize, and these chromosomes come in single nucleus after fertilization. Okay, so these two chromosome from father and chromosome from mother, they are called homologous chromosome. Okay, after DNA replication, the two finger of chromosome that we see, these are called sister chromatid. But the chromosome that we have gathered from father and chromosome that we have gathered from mother, they are called homologous chromosome to each other. Okay, so here we'll say that if a cell has to go through a cell division phase, let's say my skin has to go to divide, first of all of their chromosome. So in case of my system, I have 46 chromosome, or it's insightful to say that we have 23 chromosome, 23 pair chromosome. So 23 chromosome I have gathered from my mother, and 23 chromosome I have gathered from my father, and each chromosome are homologous chromosome for each other. Like chromosome 1 from mother and chromosome 1 for father are the homologous, are in homologous relationship. Then chromosome 2 from mother and chromosome 2 from my father, they are homologous chromosome. But and if a cell has to go through division, each chromosome, doesn't matter whether it is from mother or from father, they will divide because of DNA replication and form two of each other, and they are called sister chromatid. Now what happens during the cell division, when DNA has taken the form of chromosome, using some again various protein machinery, spindle fibers, centrosome, and a lot many protein workers inside the cell, what happens if my one cell has decided to become two cell, then these sister chromatids will be separated from each other. Means if your cell skin cells decided to become two, let's say growth hormone forms in your pituitary, it comes, it sits on your skin cells, and skin cells have decided to become two from one, then what would happen? Your cell, your your skin cells have some again signaling, and then first replication happens, and then when they will form chromosome. Every chromosome has two sister chromatid, like all of your 46 chromosome has two sister chromatid, and these two sister chromatid will be separated from each other, and new membrane system will form around these sister chromatid, and now each cells have one chromosome for all the 46 chromosomes. So after cell division of your skin cells, you have 46 chromosome in one cell and 46 chromosome in another cell. What happened basically? The sister chromatid divided. The sister chromatid, one sister chromatid went in one cell and another sister chromatid went in another cell. And this process is called mitosis. So whenever a division happens in somatic cells, like my skin cells, my eye cells, my lips, my ear, my larynx, my heart, my kidney, anywhere, so these are the somatic cells, and these cells divide by mitosis, where division of sister chromatids happen. Now one more thing, that nature have made this process of reproduction, production of new cells. If it is happening in your body, then it has to happen in a way that your sister DNA replication would happen, and same amount of DNA can be divided into two different cells using fragmentation of sister chromatid. One sister chromatid will go in one cell, another in another cell, and in the same way 46 chromatid will be separated, and they are the new chromatid chromosome now. And further, if they will divide, they will again do the same thing. But for a new organism to form, what happens if I am a sexually reproducing organism? Then my genetic cells will form a cell that will have only 23 chromosome. Instead of 23 pair, it will have 23 chromosome. Why? Because it has to meet another cell which has another 23 chromosome, and they would fertilize with each other and form 23 pair of chromosome, that we call. And these 23 pair chromosome from my body and 23 chromosome from future female body, they would just meet with each other, and they are homologous chromosome. So just imagine the homologous chromosome that will present in the cells of my son or daughter. So one partner is here, and who will be the homologous chromosome for my son? ये करोड़ रुपए का प्रश्न है भाई. So having said that, so what happens in meiosis? How do 46 chromosomes becomes 23 chromosomes? So essentially you must have understood that when meiosis happens, then essentially the segregation of homologous chromosomes happen. So every cells of my body has 23 pair chromosome, 23 chromosome from my mother and 23 chromosome from my father, and these are the homologous chromosome. When it has to, let's say if I'm talking about division of cell, then sister chromatid will separate after DNA synthesis in DNA replication in a space of cell cycle. These sister chromatid will form, and they will separate and form new cells, and they will have again the same thing. But if I'm talking about my genetic cells, then for the formation of sperm, meiosis has to happen, because here I do not want nature, my wish doesn't work. It's the nature who designed me. It's the nature who has designed me using evolution engineering. So what happens in the process of meiosis? First thing will happen, that homologous chromosome will be separated. Means every cells after meiosis 1, every cells will have 2 chromosomes. Every cells will have 23 chromosomes. Okay, 23 my parental chromosome, 23 my maternal chromosome. However, I don't think they are so much. I get it. So and then what happens? Then again in meiosis 2 process, meiosis is again meiosis 1 and meiosis 2. Sister chromatids will be separated. Now at the end of meiosis, you will have 4 cells, and all these 4 cells have same kind of chromosome, but haploid. Every cell will have only 23 chromosomes. But after mitosis, my every cells will have 46 chromosome. Why? Because in mitosis only sister chromatids divide. However, in meiosis, first homologous chromosome divides and then sister chromatids also divide, and hence you will have 4 cells each with 23 chromosomes. So this is the dynamics of DNA while cell division. How do they go from one organism to another organism by the process of this segregation of DNA, and all these things are done by protein molecules. So thinking about this nature's design. Nature's design are very complicated. If you ask an engineer that I have to bring electricity from place A to place B, what would an engineer think? Engineer would think that let's make a straight wire from point A to point B. But nature and let's say that tomorrow you want that engineer that to make another connection. So what would engineer do? The engineer would just bring this connection out and they will connect here to here. And let's say the day after tomorrow you want, I want connection from point A to point C. Then he would make another connection. He'll just take out the wire and they'll put it here. Let's say you have required. But nature doesn't break anything. If you look at nature's design, so starting from the evolution when prokaryotes first formed, and then eukaryotes formed, and then 3 branches came out, some plants formed, some fungus formed, and then some animals formed, and some, when I say formed, it took million of years to evolve all these things. So looking at the evolution of animal system, there was first porifera, and then it was some evolution happens. Then other lower organisms formed, annelids, arthropod, mollusk, and platyhelminthes even just after the porifera, and these things kept on evolving. Multicellular organisms formed, and till now we are here. And one thing, I'll give you one example to make you understand that how does nature do the designing, and how does an engineer do the designing, and what's the difference between them. As I told you that if you have to, if after let's say after 10,000, after let's say 100 years, if this building will no more be in this condition, and administration out here want to modify this building, they would just simply break. The engineer would decide to break things which are not needed, and they will just modify the room in a way that suits our need. Okay, they will just, if some bulb is not working, they'll just throw it out. If some wire connection is not needed, they'll just take out that wire connection, they'll form new connection. But nature works in an opposite way. Let me give you an example. So you must be aware of a nerve called vagus nerve. When I say nerve is a collection of axon. Okay, and every nerve has its cytone in the brain, and then axon keeps on going here and there. So a part of vagus nerve is pharyngeal nerves. So pharyngeal nerves essentially has to go from here to here in the pharynx, here to here. But so what happened from here to here? During the course of evolution, if you look at early organism, then this part is somehow located at this side, and they have so they were like, we have this vertebral column in dorsal side, and there are some which are ventral creatures, organisms. So in the beginning, nature has designed for this connection to go from here to here. For them I'm talking about. It's very difficult to make you understand this showing my body, hence I'll not show it here. So let's say this is a small early simple organism, and from here to here they have to go through heart. Okay, so heart is there, and this connection has to go through heart. Now things evolved, organisms evolved, and we evolved, And today also, if we look at our vagus, this pharyngeal nerves, the only thing that it came here, okay. Now from in lower organism, it has to go through this to this place, okay. And then through the course of evolution, these 2 points reached here. If it would have been an engineer, then engineer would have been cut this connection of nerve, and they would have made a connection here to here. It's a small distance part. But you know what did nature do? Nature never deletes its creation. What did it do? It just still going through all the way through heart, and then it is coming here. Okay. So if you ask me, my pharyngeal nerves, it starts from here, it goes all the way to my heart, and then sorry, heart is this side. So it starts from here, it goes all the way to my heart, and then come back, and then reach here. So in the same way, why do you find nature and natural design very complicated? If you look at cell signaling, if you look at all the protein molecules that are working inside the cell, so there are multiple partners existing inside the cell. But there is an advantage of this much complex nature's design as compared to engineer's design. That more the complexity, more the stability. If you think that in terms of let's consider a jungle. So if food wave in jungle is more complex, there are multiple trophic levels and there are multiple organisms, species present in each trophic level. It will make this food wave very complicated, very complex, and more complex food wave, more stable the ecosystem will be. And it applies everywhere. If you are in a more complex system where there are multiple partners working here and there to perform a simple work, you have a series of protein molecules assigned to do small works. This complexity makes this process more stable, makes the nature's design more stable, and perhaps, I don't know enough about it, but perhaps this is the reason for stable evolution of organism till date. There are a lot of good argument about nature's design and engineer's design, but the basic criteria that you have to believe on to understand and to think about nature's design without going in the creating difference between non-living and living is you have to believe on evolution and natural selection. Thank you.